Hello, and a very warm welcome to everyone joining us for today's Select Science webinar, Identification of Protein Aggregation through UV Visible Spectroscopy, the use of absorption and diffuse reflection techniques. My name is Dora Wells, and I'll be moderating today's presentation. I'm delighted to be joined by Dr. Jennifer MP, Application Scientist at Thermo Fisher Scientific. After the presentations, we'll move on to our question and answer session. Please feel free to ask any questions for the Q&A session at any time during the webinar. You can submit your questions at the left of your screen. Just click on the purple tab. Now, without further delay, I would like to hand over to Jennifer for today's presentation, and I would like to thank her for presenting to us today. Please go ahead, Jennifer.
Thank you so much for that kind introduction. My name is Jennifer Empey, and I work with Thermo Fisher Scientific as an application scientist supporting the Thermo Scientific Core UV visible spectroscopy instruments. And today I'll be talking about how you can identify protein aggregation using UV visible spectroscopy. So a little bit of background into how we can identify this protein aggregation and understand it better. The protein aggregation occurs when we have these dispersed proteins in solution, and they undergo some type of perturbation, such as changes in heat or changes in the ionic strength of the solution. And these perturbations cause the proteins to misfold or denature in solution. And then when they come together, they end up aggregating into these smaller to large particulates in solution. And oftentimes, this is an irreversible process. So once these aggregates formed, they can't be removed and they are insoluble. Now, in the body, the presence of aggregated proteins has been linked to Parkinson's disease, Alzheimer's, and sickle cell anemia, among other diseases. But if we think more of a pharmaceutical perspective, protein therapeutics can see product yield losses with the presence of these aggregates in solution. And these product yield losses can also further produce efficacy reduction losses. And if we consider a food industry perspective, the proteins in solution result in these organoleptic properties, or the taste or smell of the material that's being produced. It can also influence digestibility, and having these aggregated proteins in solution can influence these different processes for that material. Now, this is fairly common for proteins which are unstable in solution. So in any of these fields where you're using a protein, such as, let's say, monoclonal antibodies for pharmaceuticals, you can observe these protein aggregates forming fairly regularly. So it's important that you're able to recognize when these proteins aggregate in solution early on to be able to either remove them or identify that this may be something to be purified down the line. Now, oftentimes, people have used size exclusion chromatography as a method for separating out the free proteins in solution from the protein aggregates, and it's fairly effective. However, it is a time-consuming method. So the example I show here is a 30-minute time per sample, and sample retrieval after the measurement is completed can be very difficult. So others have moved to a more indirect method for identification using UV visible absorption spectroscopy to indicate that aggregates are present within their sample. So if we consider a typical UV-vis absorption experiment, we have our light source, which will allow light to be shined onto our sample, depicted by the cuvette here. And some of that light will be absorbed by the sample and attenuated. And this attenuated light is what is collected by the detector. Now, if we see in this cuvette, we have particles in solution, which are between 5 and 10 or greater nanometers in size. What can happen is, instead of this light being transmitted through, some of it can be scattered off of the particle. And what this will result in is a formation of this scattering artifact in the absorption spectrum, where it looks as if we have some absorption in a region of the spectrum. So here I'm indicating the 320 to 340 nanometer region, where our sample does not normally absorb. And that can be influenced across the entire spectrum as well. So this technique is fairly useful, as it's a quick measurement, so between 2 to 3 minutes per sample, and it's non-destructive in nature, which allows us to recollect the sample after the measurement quickly and be able to use it in further processes down the line. So for the remainder of this talk, I'll be focusing on using uv-vis spectroscopy to analyze these aggregates. And specifically, I'll be using the Thermoscientific Evolution 1+ UV visible spectrophotometer. So I chose to study bovine gamma globulin, which is a fairly stable protein in solution. And I used a concentration of 0.55 milligrams per milliliter for both my control and the solution that I would like to induce aggregation in. And this time I'm using an ionic strength change to induce that aggregation. So I'm using 2.65 molar sodium chloride in my solution to cause that aggregation to occur. And as you can see from these images here, it appears as if there is little to no scattering present. So these samples are optically clear. But when we take the absorption spectrum, we can see a fairly marked change between the samples with and without salt added. And what you can see here is in this tailing region, we have that absorption or that scattering artifact as described earlier. So this appears to influence the entirety of the spectrum as well, and allows us to understand that we can see that aggregation effect, even though our naked eye cannot see it in the sample. But what's important to point out is because this scattering artifact influences the entirety of the spectrum, we can't use the measured absorption here to determine a concentration of our free protein in solution. We need to first correct for that scattering artifact before we can determine concentration. So one method that people have used to determine what the true corrected absorption spectrum should be is this baseline correction or baseline subtraction method. So the idea here is that you take a region of your spectrum where you know your sample does not absorb. So in this case, between 330 and 350 nanometer. You average the apparent absorption measured for your scattering sample between those points, and subtract that average from each point of your measured spectrum. In this case, we're able to see that the baseline appears to correct. However, in the region where we absorb, between 270 and 290, for example here, it appears that this green curve, the baseline subtracted curve, is higher in absorption than our control. This is counterintuitive, as the control should contain more free proteins than the aggregated sample should. So we know that this method is not appropriate for correcting for scatter. And this is because the scatter is not actually the same across the entirety of the UV visible spectrum. So as you can see in this estimate shown here, the scatter that occurs within the longer wavelength region is much less than the scatter as you go further into the UV. And it can be estimated with this estimated scatter equation shown here, which is based on a Rayleigh scatter equation. And as you can see, here we would have to fit the scaling factor and offset, and it has a strong correlation with the wavelength in which you are observing your sample. So when I apply the scatter function and fit to match the baseline region between 310 and 350, where again, I know my sample will not absorb, I can fit fairly well to that baseline and subtract that corrected scatter from the measured absorption spectrum for this scattering solution. And when I do this correction, I now have an absorption spectrum that appears to have a slightly lower absorption than the control, which matches expectation, as some of the proteins would have contributed to the aggregation. So now I know that I can use this scatter correction method to produce a spectrum where I can determine a more accurate concentration for the free proteins that are left over. And using Beer's law, I was able to determine that for this sample, the concentration of non-aggregated bovine gamma globulin was 0.54 milligrams per milliliter, which implies that the concentration of aggregated bovine gamma globulin, or proteins which produce the aggregation, was 0.01 milligrams per milliliter. So next, I wanted to induce aggregation within the bovine gamma globulin sample using an alternative method. So here I'm using heat to allow these proteins to unfold, and when they come back together, they will form these aggregates. And I'm using the same concentration of bovine gamma globulin as before, and now I have no sodium chloride in either of my samples, but my other sample now has been heated to 75 degrees Celsius for 60 minutes. And now we can see that there's a clear change by eye in this sample. So now we know we have clear evidence of scattering. And this is reflected in the measured absorption spectrum of the sample, in which in this tail region, where again, I should not see any absorption from bovine gamma globulin, I see a fairly large absorption artifact or scattering artifact. And the scattering artifact implies that we do have a higher concentration of particles in solution. However, because this absorption value is so high, it implies that we're unable to observe more than roughly 10% or so of the light that's interacting with the sample, implying that it's likely going to be difficult to try that fitting method I showed earlier with these samples that scatter light so highly. So instead, to more correctly fix the scattering artifact within this sample, we have to use an integrating sphere in place. So in our integrating sphere, we first shine light on our scattering sample. That scattered light is collected by our integrating sphere, which is coated with a special reflective material. That reflective material allows the light to be diffusely reflected enough times around the sphere to essentially cancel out that scattering effect so the detector can collect the light properly. And what's important is we're able to quickly switch between a traditional absorption cell holder and the integrating sphere fairly rapidly. So this measurement can be done same day as a typical absorption measurement. So just a reminder of what the absorption spectrum for the sample looks like. When we do use the integrating sphere, because it's not a direct absorption measurement, we are unable to collect the absorption data in this manner and instead need to collect a Kubelka-Munck signal, which can be described by the equation up here. And what you can notice with this spectrum is that we have now corrected for that scatter and see virtually no to little scatter in the region where we saw a significant amount of scatter previously. Now, because we're using a Kubelka-Munck signal rather than absorbance, we can't use Beer's law and the amplitude of this peak to determine what the concentration of our material is. However, if we look at the equation up here, that signal is correlated to both the absorption coefficient of the material as well as a scattering coefficient. And as it's proportional to this absorption coefficient, we know that this signal is going to be proportional to absorbance, which will in turn make it proportional to concentration. So to determine the true concentration of the non-aggregated bovine gamma globulin in solution, I took a spectrum of the sample using the integrating sphere in which I know that I have no aggregates present. So I should have a known concentration, and I can collect that Kubelka-Munck signal from this, as well as the sample where I have an unknown concentration. And given that we again know that this is proportional to absorption, and absorption, according to Beer's law, is equivalent to concentration times path length times molar absorptivity, we can infer that we have a relation in which the collected Kubelka-Munck signal is equivalent to concentration times some constant that is uniform between the two samples. So we can then create a system of equations where we use the measured values at a specific wavelength that we choose, times the concentration of our known sample to determine the concentration of our unknown. And in this case, I received a value of 0.2 milligrams per milliliter, but I wanted to ensure that this method made sense and was correct. So I then took my heated sample that contains the aggregates and filtered it using a syringe filter to obtain a filtrate which would contain no aggregates. And in this case, I was able to take an absorption spectrum of that resulting filtrate and obtained a concentration using Beer's law and the absorption collected of 0.2 milligrams per milliliter, confirming that this method did work for the material I was studying. So we were able to determine for this sample that there was 0.2 milligrams per milliliter of non-aggregated bovine gamma globulin, which implies that 0.35 milligrams per milliliter of protein contributed to the aggregates present in the sample. Now, this correction process, be it for samples with low concentration or high concentration of aggregates, is fairly useful, especially for industries in which you need an accurate concentration for your measurements and can help you prepare for downstream synthesis. It's also particularly useful when analyzing samples in which filtration may not be possible, or samples in which there is a matrix that involves some particle that can scatter light, and you still need to determine the concentration. And lastly, it can be highly useful when you're identifying a sample needs to be purified further down the line or needs to be removed entirely and restarted early on in the process rather than downstream when it becomes more difficult to remove that. So overall, through these experiments, we were able to quickly identify that these contained protein aggregates through this uv-vis method. We were able to detect low concentrations of aggregates in the samples. And using an integrating sphere, we can produce a scatter-free measurement of these highly scattering solutions. And given we have a known sample that we can use for this integrating sphere accessory, we can then use the relations from the Kuhn-Munck equation to determine the concentration of that non-aggregated species. So before I go on, I just wanted to talk about a little bit of the parameters for this Evolution 1+ instrument. I studied the region between 240 and 350 nanometer, but we do allow for a wide range of wavelengths that can be analyzed between 190 and 1100 nanometers. This can be highly useful for samples which may have visible absorption. We have two different bandwidth options, and the noise for this instrument is between 0.0001 and 0.005 between 0 absorption and 2A. This is a double beam instrument, so we can allow for correction of any fluctuations from the lamp with time. And we do use a xenon flash lamp, which prevents any type of artifact from lamp switchover between UV and visible measurements. We do offer 21 CFR compliance for this instrument, as well as UP and EP compliance. And today I showed how the integrating sphere can be used fairly effectively with this instrument. However, we have a variety of other accessories that can be highly useful. For instance, we have the Peltier thermostat that allows for measurements at different temperatures, which is highly useful for melting experiments. The Smart Sipper accessory allows for flow cell type measurements within this instrument. And the linear 8-cell changer allows for multiple samples to be measured consecutively, which is very useful for the high throughput measurements that would be necessary for some applications. And with that, I would like to thank you for listening to this talk, and I'm free to take any questions.
Great, thank you for that interesting seminar, Jennifer. Now, let's move on to the question and answer session of today's webinar. The first question I have here is, what baseline value should you look for to determine if the sample has any aggregates?
Yeah, so that's a good question. It's really highly dependent on the sample that you're studying specifically. So I would first make sure you're taking a measurement of your solvent to kind of set where a normal baseline should be. And then from there, I'd say anything that's maybe 0.05 absorption units higher than that would constitute something to be concerned about in terms of aggregation.
Okay, great. Thank you for that. And the next question we have here is, will this method work to identify protein aggregates in any sample?
So this is specifically looking at solution phase aggregation effects, so samples that are suspended in solution. And if you do have any type of protein, given that it is making a particulate that's large enough to scatter light, this will be a useful method for detecting that aggregation.
Great, thank you for that answer. Another question we've got here by an attendee is, as most of the proteins are either semi-solid or solids, in order to record and observe the results, whether it's vital to dissolve in suitable solvents or diffuse reflectance mode. Kindly brief on that.
Yeah, so for these samples, I did use solution phase measurements. I would ensure that you're using a solvent that your free proteins can be dissolved in. The actual aggregates do not need to be dissolved in the sample because we are intending for it to cause that scattering effect so we can detect that. I guess the one thing that I would point out is you would want to make sure that the sample has been, I guess, shaken a little bit to make sure that the aggregates haven't really settled to the bottom if you want to make sure you can see that aggregation effect.
Okay, great. Excellent answer. Thank you for that. Another question we have is, would this method be suitable for studying peptide aggregation?
Yeah, so this method is really non-specific to the specific aggregates that you have present. So as long as the peptides can aggregate to a point where they're large enough to scatter light, you will be able to see the scattering effect. It's more so whether you have enough large aggregates to cause the scatter to occur.
Great. And can we do kinetics with this equipment?
Yes, so this equipment does have a kinetics mode. So if you wanted to run measurements through over time, that's a application you can use.
Perfect. And another question I have here is this technique specific to protein aggregates only? What about other particles?
Yeah, so like I was mentioning before, if you do have particles in solution which did not result from protein aggregation, you will still see the scattering effect. So it's more so about the presence of these larger particulates, whether they be from the proteins or from some other matrix you may have in solution that will cause this artifact to be present in your absorption spectrum.
Okay, great. Thank you. And is that it for, is that it uses for all the different types of proteins? So can this be used
Sorry, what
for,
was that?
can this be used for all the different types of proteins?
Yes. So as long as the proteins can be aggregated, then you will be able to see this effect. It's independent of the protein that you're studying.
Okay, perfect. Thank you. Is it necessary for biopharma customers to measure concentration of aggregates?
So it's unlikely that the concentration of the aggregates is something that you absolutely need to determine. It just may be helpful to know the concentration of aggregates that were made to potentially improve your process further down for future experiments. But what's more important is being able to correct for that scatter to get a true concentration of free aggregates that are in solution.
Okay, cool. Thank you. And this next question, I think the first half has been answered already, but I will ask it anyway because the second half I think hasn't been answered. So can we analyse peptide aggregation, and what are the limitations on the MW low limit?
Yes, that's a good question. So in terms of exact lower limit for the molecular weight of your peptides in solution, that I'm not sure about specifically. I think it's, again, like I mentioned before, I think it's more important whether those low molecular weight proteins or peptides come together and make a large enough particle to be able to result in that scattering effect. And that typically is between 5 and 10 nanometers on the low side for that particle size.
Okay, great. Thank you. I have another question here. Sorry, so what is the LOQ sensitivity of this method?
Yeah, so I don't have a specific LOQ right now to give you for this specific experiment. I can try to look at that data again to give you that information. We haven't really tested to see how low in of a scattering effect essentially that we can see, so how low a concentration of those aggregated proteins are present. But I will say that the noise level for this instrument is fairly low, so that does give us kind of some trustable room for being able to see some fairly low concentrations. The concentration I showed you before for the salt-induced aggregation is the lowest concentration I have used so far.
Okay, and is the Evolution 1 series of spectrometers new?
Yes, this is a new, newly launched product that we have for Thermo Fisher Scientific.
Cool. Thank you for that answer and the presentation, because I think that's all we have time for today. So thank you again for today's presentation. It was very informative. I found it very interesting. I hope the audience did too. Thank you to everyone joining us online and for your interesting questions as well. I hope that you found it a worthwhile session. If you have any other questions, please feel free to email me at editor@selectscience.net and I will follow up with your questions for Jennifer. And remember, you can also download a certificate of attendance in the Related Resources tab to the left of your screen. And if you would like to listen again to today's webinar or invite a friend to listen, it will be available to watch on demand in a few days. Goodbye. Thank you once again for joining us.